So let me just get my slides up. So as Frank mentioned, I'm Tori Poole. I did my RET2 summer project on this idea of combating climate change from the microscopic scale to the macroscopic scale. So a little bit about me. I work at a school super close to UCSB. It's probably about 10 minutes away, called Bishop Diego High School. It's a private Catholic school where for the past 5 years, I've taught biology and AP Bio. And then starting next year, I'm teaching a environmental science course. So this curriculum has actually been, I'll be using it not only in my on-level biology class, but I'll also be adapting a version of it for my environmental science course. So last summer, I worked, as Frank said, with Lewis in the Teague Team Lab. So Lewis is an eco-hydrolysis that is also a data scientist. And so last summer, I really had the opportunity to learn about a new field of science, right? So if you've never heard of what a data scientist is or what they do, don't worry, I was you exactly 1 year ago, having no idea like what lab I was being put into and how it was going to relate to my curriculum. But basically, I learned that scientists don't have to just go out into the field and work under a hood and work with chemicals. They can take data from people out in the field and they can use that data to predict and create mathematical models. And that's exactly what Lewis tasked me with last summer, of asking the question, how do you measure how much carbon a tree has? And the easy answer is cut down the tree and burn it. And then you can easily measure the carbon. But the issue with that is like, we don't want to cut down all of our trees and burn them to see how much carbon they have. And so basically what we did is he taught me an elementary course of how to use RStudio. So RStudio is basically, for those of you who don't know, it is a place that you can store a bunch of data and then run different models through it. You can write equations into it. And so that's what I did last summer. So these are two graphs that I made last summer where I took a bunch of data of the height of trees and the diameter at breast height, which basically just means like the width of a certain part of the tree. And we saw if the connection between these two measurements was more linear or more exponential. And so based off of those R-squared values, you can see that they're more exponential. And last summer, I was like, this is so much fun and all this stuff. And I'm like, but how am I applying what I'm learning this summer into actually my curriculum? And so throughout the school year, I was really thinking about my experience with Lewis and thinking about all the data and how important data was and how important what he's doing is, and the question of how do trees gather carbon, and like, is a forest a carbon source or is it a carbon sink? How is this contributing to climate change, and how is climate change contributing to how much carbon is absorbed? And so that's what really guided me into this RET2 program this summer, where I was designing my curriculum. I started at the beginning of the summer with a conversation with Lewis, and he really helped inspire me to create this microscopic to macroscopic climate change unit. So the main things that I'm focusing on in my unit is getting the students to collect their own data and work with data, and not just collect their own, but take data and interpret it. Look at graphs, look at like data that we can find online, and make conclusions, because I think that is such an important skill that the students nowadays need to have. And then the other main takeaway I took was this idea of microscopic changes impact macroscopic problems. So this is just kind of what my RET2 program led me to, was really a paradigm shift in my teaching. So, right, I've been teaching for 5 years now, and I've really taught traditionally linear. To be completely honest, I was given like a textbook and was like, can you teach this? And so I taught the textbook, you know, chapter from chapter, and we worked through it and the students learned something. But after my RET1 experience, I was like, there's so many connections because the world is much more complicated than this linear approach. And that was something Lewis talked a lot about. Like as an eco-hydrologist, you have to think about the ecosystem. Hydrolysis, you're looking at water. And then you're also like an environmental scientist, because every field right now, like environmental science is huge. It is the like big hot topic, and it is like climate change is the biggest concern of every person right now. Like it's not just a one person problem, it is a multi-person problem. And so I was like, why am I teaching this linear approach where I teach like cellular energy, and then I teach a bunch of other stuff, and then I teach ecology, and these are being taught separate of each other. So my RET experience really changed how I'm thinking about structuring my whole entire course, which I know sounds like a lot of work to change, but I'm super excited about it. So my new curriculum is starting to, for my entry-level bio, is focusing on the microscopic and the macroscopic impacts in every unit. So the unit that I created this summer is specifically a cellular energies and ecosystem unit that each topic is being taught to help support the bigger picture. So kind of right, biology is this whole field, but everything is connected to each other. Everything is related, right? Evolution relates to DNA, DNA relates to genetics, genetics relates to ecology, ecology relates to the microscopic chloroplast and mitochondria. And so that was the big inspiration for the 3 main activities I created this summer. So here's just a little summary of the next 3 activities that I'm going to be going over. And the thing that was really cool as I was creating these is I realized I'm hitting standards from a bunch of different sections. So if you notice, there's like, for those of you who don't know the standards, it's basically high school life science 2 and high school life science 1. Normally you kind of like stay within one section, and because I've started to have this paradigm shift of focusing on small picture, big picture, small picture, big picture, I'm starting to hit multiple standards in each unit. And I think in the long term, this is going to support my students where they have a deeper fundamental understanding of each of the standards. The first activity that I created goes with a Gizmo, which if you don't know what a Gizmo is, it is a paid subscription. It's from Explore Learning, but I know a lot of schools in town have it. And basically the question is, why does oxygen matter? So the goal of this Gizmo is that they're able to manipulate really simply different aspects of a plant and then see what it does to the oxygen output. And then in my first meeting with Lewis this summer, he talked to me about how the UN has determined that this decade is the decade of ecosystem restoration. And so that's going to be the intro kind of engage activity, like, hey, why do we have to restore ecosystems? Like, why can't we just plant 1 trillion trees? Why can't we just plant a bunch of trees and absorb all the carbon dioxide? So my hope is that the students start looking at this plant and seeing this oxygen changing, which is that microscopic change, but then relating it back to this global idea that we can't just plant a bunch of trees. That's really complicated and probably not going to be the best way to absorb carbon dioxide. So one of the best ways to do it would be to start restoring the ecosystems that have sadly been destroyed. So in this activity, for those of you who have never seen a Gizmo, basically the students have the ability to manipulate the temperature, the light intensity, and the CO2 level. And as they do that, if you notice here, I have a couple screenshots of the light intensity being changed. So in the first one, right, it's down low and then it goes up high. And then if you look at the oxygen flow is lower and then it goes up higher. So basically the student's goal of this activity, I have like a worksheet that leads them through, is to determine the parameters that create the most oxygen in a simulation. So how can we get the highest oxygen output? If we have the highest oxygen output, that means our rate of photosynthesis is the highest. The students at this point don't know about photosynthesis unless they learned it in 7th grade life science, which I'd say about 50% of my students come to me with that prior knowledge. And once they determine those parameters, I'm going to have them choose one to manipulate and then graph their data, right? So this is also part of Gizmo. So it's really cool. They can click on that graph button and then record, change the intensity, record, change the intensity. They could also do this with temperature or CO2 levels. So once each individual does this, they'll be able to share with each other like, hey, this is what happens to the graph when light intensity. This is what happens with temperature. This is what happens with CO2. And the hope here is that they're getting that data and then they're able to analyze it right away, right? So if you look at the data on this graph, you start to see that there's an increase and then there's a point that it levels out. So there hits a point that light intensity isn't going to increase the oxygen flow anymore. And so they'll partner up with people who did something different than them, and they'll find out, again, what's going on with CO2 and temperature. After this activity, they're going to have a little bit of direct instruction. So, right, you can't be in a high school biology class with at least a little bit of direct instruction. But part of my direct instruction has started to change into, rather than teaching these two processes as two separate things, we're really going to be teaching them together and teaching the mirrors of them. So the students are starting to make that connection of how do these microscopic processes connect to one another. And then hopefully after their next experiment, they'll be able to connect it to a bigger problem, right? The microscopic to the macroscopic. So the next lab that I created is with these algae beads. If you are a biology teacher, you need to talk to Algae Research and Supply. It is a company based out of San Diego. Their owner's name is Matt. He is awesome. I actually reached out to him and had like an hour and a half, almost long phone call with him where we worked on a Google document, and he invited me to a presentation this morning. And he wants to help teachers. So little plug for them. But basically, algae does photosynthesis and cellular respiration. And so the students are going to get these little algae that are inside of an indicator solution. And as the algae do either more respiration or more photosynthesis, the indicator is going to change based off of the pH in the background. And this takes a while. So it takes a while for the indicator color to change from like yellow, orange to the purple, blue that you see. So the students will set up their lab, they'll run it, they'll have a couple different options to choose them, giving them some voice and choice. And while they're waiting, I'll get back to that. While they're waiting, they're going to also do research and connect algae back to climate change. So they set up their experiment, have a couple different options, and then they'll individually either read. So 2 students have an article to read, and then the 3rd student has 3 videos to watch. So, right, maybe they'll want to do the videos. Some kids are more like, I'd rather just read the one article. That sounds easier. And they'll have to summarize what they learned about algae and its impact on climate change to each other. That's while their experiment is running. So you see, this is one example of an experiment they could run. They could run multiple different types of it though. So kind of my idea is light intensity, temperature, or light color. And I have a lab write-up that goes for whichever one they end up choosing. And then they're going to gather their own data. So their data that they're gathering is the pH of the indicator, which in turn tells them how much carbon dioxide is inside of the solution and what the rate of photosynthesis was. So you can see here, this is a light intensity setup. And if we look, the little algae beads that are closest to the light, we're having a higher rate of photosynthesis than the algae beads that were further away from the light. So hopefully they're understanding that there's these small changes at the microscopic level, right? The rate of photosynthesis, and that could have an impact on the environment because of the indicator, right, it's changing the amount of carbon inside the indicator. And so then they'll be able to relate what they saw in this lab, what they learned in their research, and connect it back to that idea of ecosystem restoration and climate change. This is an assignment that I actually designed earlier this year called the carbon cycle. And basically where you play a game and you follow it. It's a fairly common biology like activity, but I kind of put my own little spin on it to connect it back to that microscopic and macroscopic. So what they do in this carbon cycle game on the right is they click through the different carbon pools, and then they're told based off of a random die where to go next. And then they move to another carbon pool, or they might stay in the same carbon pool. The microscopic part is they're going to look at what does the carbon atom look like in the atmosphere, right? So it's CO2 in the atmosphere, but as it goes into the ocean, did it become bicarbonate, right? So did it get dissolved? What are the different things that we're kind of looking for? Is it in methane form? Is it in carbon dioxide form? And so they'll have the opportunity to look at that microscopic scale as well as the macroscopic carbon cycle. Then my favorite part, which is where my apartment this year became a little zoo, is when we start to put all of this together. So, right, the original idea was climate change, looking at it from the microscopic to the macroscopic, and using real data to help interpret and make predictions, just like a real data scientist would. So the plan is that the students will use their knowledge of photosynthesis and cellular respiration and all of the nutrient cycles that they've learned about previously, so that would be like before this unit, to create their own ecosystems. So here I have an example of something they might design. This, they could design so many different ways, but the idea is that they're creating a self-sustainable, closed-off ecosystem. And then they will choose what data to gather. So far, some of my examples and my ideas for data to gather would be pH of soil and water. As you can see, I have a little vernier system and the probes. Those can measure pH, temperature, carbon dioxide amount, oxygen amount. And it's going to be up to the students. Hey, what type of qualitative data or quantitative data do you want to take? And also qualitative data, like, do you want to describe the environment? How is the plant looking? How is the animal looking, right? I had a goldfish in my one side of the ecosystem, the aquatic ecosystem. Is it moving slowly? Is it moving a little more quickly? Does it react to when you get close to it? So basically the students will do all this data gathering for about 3 to 4 weeks. So this will be a long-term project that then leads into the next unit as well. So this isn't just, right, the only thing they're doing throughout the time, but you kind of have to establish a baseline. And then we want to use our baseline data to make a prediction of how your ecosystem would respond to an issue that we're facing with climate change. So what would happen if we increased carbon dioxide in your ecosystem? Or what would happen if we increased the temperature? And that way these students are really getting the idea that these processes, right, the mitochondria and the chloroplasts are working really hard to do this stuff that's keeping all the animals and plants alive. But the environment can change that small thing and impact it from working correctly, right? Like if we hit too high of a temperature and we get to a point where proteins start denaturizing, like that could be really bad. And there are actually some areas, I was talking with Matt from Algae Research and Supply, and he was like, there are some areas that are expected to hit that temperature, you know, pretty soon. So like, how are we dealing with this? What is that looking like? And so hopefully hopefully by the end of this, my students feel comfortable in understanding that climate change is a real serious problem, but there's not a really simple solution, but we can use models to help us make predictions. And models are just like so important, and gathering data to support why we think our model is going to react a certain way is so important. With that, so those were the 3 things that I designed. For this summer. I don't know what just happened there. But those are the 3 things that I designed for this summer. And my last slide was a thank you. So I had a picture of Lewis and the Teague team on there. Thank you so much, Lewis. Like, our conversation at the beginning of the summer is what inspired this whole thing. And hopefully in the future, I have a bunch of little data scientists graduating from my high school, moving on to college. And then, you know, you can work with them then. And thank you to Barbara, Frank, and Marilyn for just all your support this summer and helping and coming up with some new ideas. And then finally, thank you to the algae research and support. They have been awesome. And if you're biology or even chemistry, reach out to them. He really wants to help teachers talk about climate change. So I don't know what happened to that last slide, but there we go. Thanks, everyone. If you have any questions, let me know.